All right, so it's inspiring for me to be here to introduce this event to celebrate your accomplishments and your talents. This is a very unusual talk compared to the research talks I normally give, so it actually took me much longer to think about and prepare this talk than my 40-minute research talk I'll be giving this afternoon, or really than probably any talk I've given since my tenure lecture many, many years ago. So my name is David Liu. I was asked to reflect on my career as a chemistry professor and give advice to the talented 12. And then I sort of looked into your bios and was incredibly humbled and realized that you're more likely to give me advice than the other way around. And then I realized that being asked to reflect on my life and give advice to you makes me feel very old. And then I had a sobering moment when I realized that this is actually the 20th year I've been a professor of chemistry at Harvard. And then I realized I really am old. So this is my effort to try to distill my experiences into some thoughts that hopefully might be useful at some point to each of you. So I grew up in Riverside, California, went to a public high school there called Riverside Poly High. Became interested in science really throughout my childhood. I think from playing with insects, to put it euphemistically, in my backyard when I was a child, blowing stuff up in my front yard, basically doing all sorts of things that made me curious about the universe and especially about the natural world. I went to Harvard as an undergraduate, graduating in 1994. Working in the labs of E.J. Corey, where I had my first taste of experimental chemistry. E.J. Corey, of course, is a luminary synthetic organic chemist, but he had a small biological chemistry, molecular biology sort of subgroup in his lab at the time, and I had the pleasure and the mind-expanding opportunity to work in that subgroup on some chemical synthesis, but really on trying to tease apart the mechanism of a remarkable enzyme, oxidosqualene cyclase, the enzyme that turns the long, floppy chain epoxide into the classic steroid tetracyclic ring system. That experience really opened my eyes to experimental chemistry and the interface of organic chemistry and biology. I then did my PhD with Peter Schultz at Berkeley, working on efforts to expand the genetic code in cells, and then graduated in 1999. And for those of you who weren't 3 years old in 1999, this was sort of the height of what Alan Greenspan would famously call the irrational exuberance period. So institutions were flush with money. People were hiring people like crazy. I think I was the beneficiary of that irrational exuberance. Because towards the end of my graduate career, I began to get invitations to give seminars, which I eventually would realize were sort of unsolicited opportunities to interview for assistant professorships, and ended up starting my assistant professorship the same year in 1999 that I got my PhD, starting at Harvard. So that explains why I've been a professor for 19 plus years now and have all the age and cynicism of somebody who's been a professor for 20 years, but still feel like I have a lot of science hopefully ahead. Okay, so what is some advice that I can pass on to this astounding group of young people? I tried to distill it down to messages starting with professional, going increasingly personal. And my first piece of advice is that it's really all about the people. Your research group or your company's success, your own professional success will be primarily dependent on the talents and the dedication of the people around you. And it took me a while to actually realize just how critical the people around you are, especially in the scientific enterprise these days. And one of the aspects of one's coworkers that I think is critical is their diversity. So while it's almost cliche these days to say that a diverse workplace is beneficial, I've really lived through periods where our research group, a somewhat outdated picture of which is shown here, has been more diverse or less diverse. And there was a time, maybe 9 years ago or so, when our research group was disappointingly homogeneous. We had fewer than 10% women in the group, and even the group members that we had, men or women, tended to be similar in the way that they thought, and that created an environment that was not as healthy and intellectually fertile as the environment that I currently enjoy in my lab. Hypotheses that turned out to be incredibly wrong. I think we pursued in part because we all reinforced each other's thinking. Now one of the aspects of my group that I treasure the most is the fact that it's so diverse. So not just in gender, but also in background, in ethnicity, in orientation, just about every aspect that can influence somebody's perspective contributes to this diversity of proposed ideas and solutions and questions. And of course, hand in hand with creating a diverse group comes with making sure that the environment that you create in your workplace welcomes the diverse perspectives that such a diverse group will bring. And that's been enormously valuable over the past years. Okay, so second, I think especially in academia, maybe a little bit less so in industry, but I suspect in just about all walks of professional science life, there is a tendency to implicitly or sometimes explicitly try to convey to our trainees that their highest goal, their highest aspiration should be to grow up to have just the same profession that we have. In other words, to clone ourselves. And I think that's an unhelpful and really unhealthy perspective. Instead, I think our highest obligation is to serve as a matchmaker, both of the science that we do and of the careers that our trainees hope to launch. So figure out what the strengths and weaknesses of your coworkers are, match those strengths and weaknesses with specific scientific activities, and match their career aspirations with specific steps during their training that will help them reach those goals. And therefore, of course, some fruitful futures for your trainees will likely have nothing to do with your present career. It's something that I think we don't emphasize enough to our trainees. Exhibit A for that, since she's in the front row, I'll embarrass her a little bit. Nicole Gao is one of this year's talented 12. She led a, I think, really interesting piece of science during her postdoc in my group. She engineered and evolved a molecular machine, a single polypeptide chain, that is programmed by a piece of RNA and that can target an individual base pair in the genome, the vast genome of a living cell, including a human cell, and can change an AT base pair to a GC base pair. And because those point mutations, especially in that direction, changing an AT base pair to a GC base pair, happen to correct a large fraction of pathogenic human point mutations that cause genetic disease, this so-called adenine base editor that Nicole developed is being already widely used both to illuminate biology and to validate potential treatments for human genetic disease. Now, Nicole's graduate research with Craig Townsend, luminary bioorganic chemist at Johns Hopkins, ended in a first author Nature paper in which she disclosed the surprising mechanistic details of the biosynthesis of a certain class of monocyclic beta-lactams. Her postdoc ended with a first author Nature paper in which she reported this work. So as you might imagine, she began to be courted by, and I began to receive inquiries into her potential availability to start as an assistant professor and launch her own academic career. But Nicole realized that her aspirations, her personal qualities, her priorities were best served by trying to connect this technology that she developed to benefit human patients. She became very passionate about that. aspiration, and so she decided to join a biotech company as a research scientist, as one of the first bench scientists. Beam Therapeutics, founded to translate base editing technologies, including the one that she developed, into cures or treatments for human genetic diseases. So I take Nicole's story as a great example that how accomplished you are in terms of your publication record and the achievements of your scientific endeavors does not necessarily mean that you should strive to become an academic, for example. In her case, I think this was the perfect choice for her, and she's already had a major impact on the industrial side and therefore on the patient side of these technologies. Okay, third, I think everybody's aware that problem selection is critical, but I thought I'd take a couple minutes to go over some aspects of problem selection that have helped guide me over the past couple decades. I think the argument for how to think about problem selection is that you all, by virtue of your gifts and your talents, are in this amazing position to impact society. And because life is so short, I think you have to hold yourself to a very high bar, almost a ridiculously stringent bar, with respect to how important your problems need to be that you choose to work on. So what's a good litmus test for that? Well, I like to tell my students to choose problems that pass the mom test. I have very few photos, it turns out, on my computer of my mom. This is one of the few ones. In fact, this is one of the few photos I have of my immediate family, which might explain why instead of my dad being in the photo, there appears to be a toilet in the background. I suspect my dad was actually taking this picture. But the mom test is you should be able to explain to your presumably non-chemist mom in 1 or 2 sentences with maybe a maximum of 1 semicolon why each project you're working on is important. And if you can't explain it to them in 1 sentence or 2 sentences, then you should either rethink how you're explaining the significance of your problem, or perhaps you should think about working on a more important problem. I certainly had, during my academic career, projects which I think failed the mom test, and repeatedly trying to explain to lay people how the problem that I chose would impact society was very helpful in guiding how to adjust those problems and, in some cases, choose new ones. Fourth, I think it's critical to nourish collaborations and connections to as many other fields as possible. Everybody who applies for research funding is asked to list their key publications. This is one of the things NIH asks periodically. I did this recently, and that publication list is shown here, the one that I submitted at least. One feature of this list that I was struck by is the fact that every project every paper on this list really came from a marriage of multiple fields, starting from our very first papers that tried to marry organic chemistry and molecular biology, something that's much more common now. But along the way, there were some moments that, for me, were very informative and expansive in shaping the nature of our research. For example, in 2014, we published a paper in which we integrated organic and medicinal chemistry with X-ray crystallography, thanks to Marcus Seeliger's lab at Stony Brook University, with animal physiology, thanks to Malcolm Leistering at Irvine and Maureen Sharon at Albert Einstein School of Medicine, with metabolism and diabetes, thanks to Alan Sagatelian at the Salk Institute now, Wei Jin Tan at Chicago contributed to protein purification that led to a crystal structure, all part of this highly multidisciplinary project in which we used DNA-encoded libraries and in vitro selection, X-ray crystallography, and a lot of animal studies to identify compounds that could inhibit the enzyme that degrades insulin in an animal. In 2016, we published this paper that integrates protein evolution with agriculture and insect biology, working with Tom Malvar and Victor Gusov and their coworkers at Monsanto. And I never would have thought that our adventures in protein evolution would lead to projects in killing insects. But that was really an eye-opening experience. And of course, we've had a number of studies recently that in the genome editing space, one of which shown here was a 5 or 6 PI collaboration integrating not just molecular biology and genome editing, but also animal physiology, electrophysiology, and the biology of hearing, combining our lab's efforts with those of a number of faculty at Mass Eye and Ear, including Charlie Lieberman, Jeff Holt, and Zhenyi Chen. And all of these collaborations, all of these connections that we made to other fields launched a number of additional projects. And I think that experience of being a contemporary chemist and having the opportunity to access information from and to transmit information to so many other researchers in neighboring disciplines has proven to be one of the most enabling features of current 2018 chemistry. And I hope everybody in the Talented 12 takes advantage of that situation. Okay, so fifth, you'll all be evaluated, no matter what your profession, periodically. And especially early on when you get evaluations from journals, from grant agencies, funding agencies, from your boss, I think they can elicit a strong emotional reaction. Here's an NIH review from one of my first R01 proposals on DNA templated synthesis. I won't read the whole thing, but the key is the goal of this very innovative but unnecessarily heroic revised proposal is to couple nucleic acid evolution with organic synthesis. Although the reviewers indicated there was much to like about this proposal, they also mentioned it was extremely hard to read, and at no point was there a simple, clear statement made that describes the conceptual, strategic, and most especially the tactical issues associated with the proposal. When I read this, I was pretty sad because I had spent an enormous amount of effort, months actually, writing this revised proposal and had convinced myself, of course incorrectly, that getting this R01 revision funded would make or break my career. And it wasn't funded. But there were a number of messages just from this paragraph that proved to be incredibly useful in all of my subsequent grant writing experience. The most important of which are these two words, unnecessarily heroic. Until that point, I really thought that the more preliminary results, the more data, the more stuff you could put into a proposal supporting your ideas, the better. That turns out to be probably more the opposite of true than true. In reality, what's really important for communicating your science, both in papers and in grant proposals, is to provide a very clear, boiled down rationale for why what you propose is meritorious, is impactful. And then it's almost, beyond that, the game is almost to write as much of a haiku as you can, to cut out everything else and minimize the opportunities that your reviewers will have to both get confused and to find fault with what you propose. So this was really a wake-up call for me because the words unnecessarily heroic really captured that the effort I had spent was really effort spent in exactly the wrong direction and shaped all of my proposals from that point on. That said, not everything that you get, not all the feedback you get from your evaluations will necessarily be something that you can take constructively. And so my next piece of advice is don't let the rest bother you. So also in this proposal was an idea that I thought was pretty cool. The idea was we could, so organic chemists have traditionally discovered chemical reactions by focusing on a specific set of substrates and an anticipated pathway that they think those substrates might react under, and then exploring different reaction conditions to study whether those substrates indeed can undergo a reaction. We proposed taking a different approach in which we would instead link dozens of different functional groups to individual DNA sequences and subject them to Darwinian selection that was specially set up so that if the substrates underwent a bond-forming reaction, the DNA sequences that encoded those two substrates would amplify themselves. And then we could read out those DNA sequences. Back then, we didn't have high-throughput sequencers, so instead we read them out by DNA microarrays. And using this technology, the greenness of a spot on the microarray would tell you what has reacted with what. Okay, so we even had preliminary results showing that in the presence of palladium-2, you could get certain reactions to take place, and this was the feedback we got. AIM-2 is now seeking to develop new reactions by mixing lots of things together and seeing what reacts. This seems highly speculative and perhaps contrived. Either you mix things together that can react, in which case they will react, or you mix things together that don't react, and they probably won't react. So that was the feedback I got. It's a little bit of a head scratcher. This is the entire feedback I got about this AIM, and so my advice is to not let the rest bother you. We ended up discovering a few interesting reactions using this approach, which are summarized here. And also, I think in one of the more heartwarming aspects of that line of research, a number of now very prominent, well-known reaction-discovering organic chemists actually told me that this approach influenced their own efforts to take a broader substrate-independent approach to reaction discovery. So in other words, minimize the amount of time and energy that you waste on matters that you have no control over. Which actually is related to this next point, which is to find peace in self-selection. Of course, our laboratory is really organized around the principles of biological evolution, so it might make sense that I've come to have a real reverence for natural selection. And I think that reverence applies to some aspects of one's professional life as well. The quality of your professional life is going to be very dependent on the overlap between your aspirations and your environment's appreciation of your goals. And that simple realization, I think, is liberating because it makes you realize that you really shouldn't focus on tenure or the equivalent of tenure. The most important advice I can give you about how to find peace in your professional life is to do your science your way. Tenure is, I think, a pretty archaic system, conceived with the idea that it gives academics the freedom to research ideas that are controversial or unprecedented. But as chemists, you have something that's even better protection than getting fired from your job that allows you to work on ideas that are unprecedented or controversial. It's called data. You can actually generate results that demonstrate to people that your ideas are actually worthy of being pursued. Okay. And then finally, almost finally, I think it's important to maintain work-life balance. And I've qualified that statement with macroscopic work-life balance because I think all of you, all of us have gone through periods, some quite important, where our work-life balance is out of whack. And I think that's actually okay as long as it doesn't become sort of the overriding organizational principle of your life. So we have all worked for weeks or even months where pretty much all we do is drive towards one goal. But in the end, I've personally found that maintaining a set of hobbies, maintaining a connection to other aspects of my life outside of chemistry is critical. So for me, most of my hobbies are at the intersection of art and science and being a nerd. I enjoy doing wood turning and a lot of photography. This is a sketch of my daughter that hangs in my office so that I can remember that while chemistry is life, life is a lot more than chemistry. This is an interesting sort of shadow sculpture. So this is a portrait of my family that I made inspired by an artist, Kumi Yamashita, made from stacks of kids' wooden letters, different heights. It's illuminated by a point source of LED, and it just creates a shadow that looks like my family. And this is a sort of original piece of electronic art. One of the most famous cellular automata is the game of life. And this is a display. It's a few feet by a few feet that plays the game of life and is programmed to look like cells. The LEDs cast on doubly frosted plexiglass, so there's actually a lipid bilayer that surrounds each cell, and they fade in and out as they grow and die. So doing things like this has really helped give me perspective and keep me mentally fresh for the rest of my work. Another aspect of my work-life balance that has been unique is, as was recently noted by a reporter, I disclosed that for about 4 years I ran a card counting team, a blackjack team, made of Harvard undergraduates and grad students. This was actually a class that I taught every week for about 4 years, just as a separate voluntary class. The attendance in the class was extremely high, much higher than in my science classes. And we developed our own math systems to count cards and to be able to beat just about any game of casino blackjack. We had 2 systems, the easiest of which is actually encoded in this tree. This was a tree that we put on T-shirts for the team, and the color and the number of leaves, the number of points on each leaf actually correspond to numbers. And this tree, if you were to memorize all this information, actually tells you how to make all of the blackjack decisions against any dealer up card using the first of these systems. And so for 4 years, every 2 or 3 months, we would actually fly, usually to Las Vegas, and put into action absurd sums of money. And it was an amazing experience. Unlike any kind of teaching experience I had before, the team was always small, 14 people total, but at no time was the team larger than about 7 people. And just having that experience of being able to teach a small group of people something that they were really enthusiastic about was inspiring, was amazing. And I still remember vividly, we would often return on the red eye from Vegas to Boston, JetBlue, flight 777, by the way. I would step off the plane on the red eye Monday morning and then roll into my morning class and teach it. And I was pretty tired at that point. Sometimes we were up early into the morning playing. And at several moments I would sort of ask myself, why am I doing this? Usually by that question I meant, why am I flying to casinos playing blackjack with students? But occasionally, after a particularly good trip, I might have meant, why am I a chemistry professor? But it really was an incredible kind of experience that reminded me, taught me a lot about millennials, first of all, and allowed me to understand the culture of generations younger than mine in some amount of depth. That's what happens when you share hotel rooms with students in Vegas. And also just served to do something that was also intellectual and required a huge amount of hard work, but was very different than the kind of chemistry research we did. Okay, and then finally, I'll just say that it's critical to give back. By virtue of your dedication and your gifts and your talents, you're all in this unique position where you'll have the opportunity to contribute even more to the sciences, to our country, to the world in general, than you were given as you grew up through this process. So I encourage you to contribute to the review of journal articles, to the review of grants, to advising the government, to participating in community outreach, to getting students involved. One of the take-home lessons for me over the past 19 plus years has been that mentoring is really a mutual gift, that I've learned at least as much from teaching 3,000 students in the classroom and exactly 100 PhD and postdocs as I've taught them, and probably the exchange has been far in my favor. Just about every aspect of communicating science I've learned from teaching. And so that leads me finally to thank a remarkable group of students, and these are the graduate students and the postdocs I've had the incredible honor of working with over the past 19 plus years. I also want to thank my mom and dad and the 14 blackjack ninjas that I trained. Their names are being withheld for their protection. And I'm grateful to these agencies for their support over the years, and thank you very much for your attention.